Okay, for class today, we touched a little bit yesterday that you would be working on writing your enzyme lab today. It's going to be inquiry based, and that means that you get to decide on which variable you would like to test the effectiveness of an enzyme on, whether you're going to work with varying temperature or pH of the enzyme solution. Learning targets are similar to yesterday. Enzymes have optimal pHs, they have optimal temperatures, and then based on your lab design, you're going to have a better understanding of how the enzyme concentration affects the action of the catalase. And then the last one, understand how a substrate, in this case, which is going to be hydrogen peroxide, affects the catalase action. When we look at the lab in terms of the inputs and outputs, the reactants and the products, we're going to be adding hydrogen peroxide to a flask, and then we will add catalase enzyme to it. When the reaction is taking place, we would have output of water and oxygen. Again, remembering that catalase is an enzyme, so it's not consumed during the chemical reaction. It just helps lower the activation energy. If you would please upload this lab, this is going to be the brainstorm and general information that you need to think about so that when we practice on Thursday and run live on Friday, you're able to collect data and give an accurate assessment of your results in terms of how the enzyme reacts when it's in optimal pH or optimal temperature. In Canvas, it's labeled today's date, 11/13/18, testing catalase activity lab sheet. I also have a hard copy for each of the lab groups, and you can grab that up by the front table where the eye wash station is. I would like one of these turned in hard copy to me before the end of the hour today, so please give it to whoever is subbing for me. When you open up the lab sheet, you're going to see the entire form at once. The first part, upper left-hand corner here, is the question that you're testing. And so you need to decide when you get in your group members, are you going to be looking how the environment in terms of the pH or temperature affects the catalase action. Decide on your independent variable then, and then dependent variable. Our actual lab design will be using the lab quests. We'll be using some oxygen sensors, and then we will have a flask that will put a dropper, a sealed stopper, and it'll measure the amount of oxygen production. And the more oxygen production, the more active the enzyme is in the given conditions that you're going to be deciding on and setting up to run for Friday. On Thursday in class, we will be working with the LabQuest interfaces. You'll be able to look at how the gas sensors work, making sure that we have our 125 milliliter flask, that it's clean. We'll also be working with some micropipettes, and then you'll practice adding the enzyme to the hydrogen peroxide, recording, and using both the iPad graphical analysis from the iPad, which we asked you to put onto your devices yesterday. Again, we plan on running live on Friday. Thursday is going to be a test. Some of the data that you're going to want to remember as you're working on this lab design here in a little bit is that we're measuring catalase activity, and we're going to be measuring it in kilopascals. Some unit conversions as you're working with the catalase is that 100 microliters is the same as 0.1 milliliters of catalase, and when we deal with 10 mLs, we're working with a 1.5% hydrogen peroxide substrate. Our typical runtime is going to be probably not more than 200 seconds in terms of time frame. We will want multiple trials, so 200 seconds would give us a pretty good visual and graph of the enzyme activity in different conditions. On your lab sheet that you have, it's showing an initial model, so if you can draw that setup, showing that you'll have a flask and a sensor, you'll have the LabQuest Pros on the table. Your iPad with the Wi-Fi and graphical analysis should be able to sync the data as it's being collected. So if we can provide a sketch here, variables, give a prediction of what you think is going to happen, and then reasoning for that. So this is where once you get into your groups, you need to talk through and decide in that prediction category, what is the optimal pH of beef catalase? What's the optimal temperature of beef catalase? Then you're going to be setting up some conditions that you'll be testing to see which environments did give the best optimal catalase action based on the amount of O2 that's produced. Write down constants, things that are kept the same. Again, we want to run a controlled experiment, so many of the variables need to be the same throughout, so that we can, when we collect our results, we know that it was the one variable that we're testing that's leading us to those results. Multiple trials, we'd like to have at least 2 trials based on each condition. We should have some type of graphical analysis. Remember, with our graphing, we need to have titles, we need to have units on the X and Y axis. We also need to label the X and Y axis, and if a key is necessary, that we have that there as well. Towards the end, our explanation, based on what you thought was going to happen, you're going to say what that claim was, based on what you know and what you researched on catalase enzyme activity. Based on the data you collect, was the claim supported with evidence? If it was not, then you need to explain what the evidence showed, and then the reasoning behind that. And then giving overall reflections and extensions. Should be a few sentences there, concluding the lab. So this is what that whole sheet is that you should already have in Notability, which we just asked you to do. I need one of these from each one of your groups by the end of the mod today. When we run on Thursday as a practice, we should be able to, you'll see here the X and Y axis, running time about 200 seconds, and then pressure again is going to be in kilopascals. We should be able to go to areas on the graph and be able to get the slope, the amount of oxygen pressure per second, which tells us a relationship to that catalase activity. This was sample data from last year, where you could see that we started the lab but didn't have the cap on the flask. Once you put the cap on the flask, you definitely can see some oxygen pressure being put inside that internal condition, and then it starts to flatten. Over here, what ended up happening is that the top popped off, and you could see that it fell back down, but that it still did have some oxygen being produced. So data is data. We'll see on Thursday as we practice running these setups, some of the trickiness of trying to make sure we're consistent in the lab setup and the design. So some guided questions that you want to think about. You need to decide your question there. What is your investigation going to be surrounded around? How does temperature affect catalase? Or how does the pH affect the catalase? How does different types of enzyme concentration affect the enzyme activity? Or how does substrate, the hydrogen peroxide concentration affect the enzyme activity? So the first two deal with environmental factors, changing temperatures and pHs, and then these deal with what happened, the 3 and 4 address what if we change the enzyme concentration or the substrate concentration. So today you're going to be picking one of those guided questions on that last slide. Create an investigation based on that lab sheet. Create a model. We should have at least 3 different temperatures, enzyme concentrations, substrates. Also think about denaturing. What's going to happen if our pH gets too high? I would have an extreme condition so you can see whether or not that claim is supported. And then make sure we have our constants labeled, your labeling graphs. Make sure you have goggles for Thursday and Friday. We do have class sets if you don't bring them. Lab groups. Group number 1, working at table 1, Maddie F., Macy, Heather, and Adriana. Lab group 2, Brooklyn, Alyssa, Jaden, and Michael. Group 3, Heidi, Lincoln, Samantha, and Dylan. And group 4, Cole, Ann, Taylor, and Caitlin. Group 5, Isabella, Brooke, Ava, and Karina. And group 6, Anna, Michaela, Tegan, and Lydia. And group 7 at table 7, Sam, Raina, and Caitlin. So these will be your group members. Again, one of those full sheets need to be filled out so you can work on them in your Notability, your electronic notebook, but I need one hard copy when you're to that point and ready to do that. Again, it's right at the front table by the eyewash station. When you finish the Catalyst Lab today, then you have Chapter 9 reading guide questions on cell respiration that you can be working on. I will see everybody on Thursday.